Hello, I'm going to talk about genes, mitosis and meiosis. I'm going to try and help you understand how researchers might identify genes that are linked to diseases. I'm also going to specifically talk about genes for freckles and gene for eye colour and gene for earlobe shape. In our system, we're going to talk about a cell which has two chromosomes. So here are the chromosomes. A pair of chromosome 1s and a pair of chromosome 2s. And here are the details about the genes we're interested in. Gene X is a candidate gene for hole in the heart. Freckles gene is present. Eye colour gene is present, all on chromosome 1. And on chromosome 2, we've got the earlobe shape gene and this gene Y, which is a candidate gene for schizophrenia. Here's the cell in interphase, and inside that nucleus we've got diploid DNA, so two pairs of each chromosome. And we have, for these 5 genes, this genotype: X1, X2, big F, little f, big B, little b, big E, little e, Y1, Y2. So the X gene is heterozygous gene with 2 different alleles, one of which may be involved in hole in the heart. The heterozygous gene for freckles, big F is normal, little f is recessive for freckles, therefore the phenotype of this heterozygous genotype, big F, little f, is no freckles. Similarly, the eye colour genotype, big B, little b, the dominant allele, big B, is for brown eyes, therefore the phenotype of big B, little b is brown eyes. Earlobe shape, the dominant allele is unattached, and although the recessive allele of attached little e is present, the genotype is unattached. And then we have heterozygous for gene Y, which may be linked to schizophrenia. So here is our cell going through mitosis in prophase. So the duplicated chromosomes condense, and you can see the chromatids for each chromosome. And the chromosomes have appeared at random positions within the nucleus. In metaphase, the chromosomes all line up in the center of the cell. In anaphase, the chromatids are pulled to opposite poles along the spindle fibers, and at telophase the cells divide. And you will notice that we have 2 identical cells with the same genotype, because that's what mitosis does. The cells are diploid and identical. So let's consider meiosis. Prophase in meiosis, the duplicated DNA is condensed as before, but this time the homologous chromosomes, in other words the 2 chromosome 1s and the 2 chromosome 2s, line up and pair up. And in metaphase 1, they line up side by side in the center of the cell, and in anaphase 1, the chromosomes with the chromatid pairs are pulled to opposite sides of the cell, and the cell divides in telophase 1. So the key here is that we've now got 2 cells with different genotypes, but they're still diploid. So there's a second round of division. Prophase 2 has the chromosomes in random positions. Metaphase 2, the chromosomes align along the center of the cell, and just like in mitosis, in anaphase 2, the chromatids separate to opposite poles of the cell, and the cells divide. Now, in this example, you can see that actually, although we've got 2 different genotypes and they're haploid, the genotypes are identical in 2 out of the 4 cells. So there's 2 pairs of identical cells, and that's not what you would expect. The reason that's not what you expect because there is another issue, and that issue is crossing over. So in prophase, in the first prophase of meiosis, you get crossing over. Therefore, chromatids on homologous chromosomes exchange material, and the DNA is copied from one to the other, and they exchange parts of the chromosome. So when this happens, the result of meiosis is that there we see some swapping over. The result in meiosis is that you will get 4 different genotypes for the haploid cells, the haploid daughter cells that are produced. So the first reason for difference of cells in meiosis is you get segregation of chromosomes. Second reason is you get crossing over in prophase, and the breakpoint's called chiasmata, and they swap over, and that allows you to get differences. Now that's important for the following reason. Looking back at this diagram of the chromosomes, you can see that there should be no link in individuals between freckles and earlobe shape. They are independent of each other because they are on different chromosomes, so they would not be inherited as a unit. As an alternative to that, there is a link between freckles and eye colour. They would often be inherited together as a pair because they're on the same chromosome, but not always. Because of crossing over, you can get inheritance of one of the genes, freckles or eye colour, without the other one. Now, in our analysis with heart disease and schizophrenia, the schizophrenia gene, the tendency to schizophrenia, is going to be associated with earlobe shape. There's going to be a common